So, introduction to hemostasis and thrombosis. My name is Dr. Euche Ezigbo. Okay. Once again, you're welcome. So, today our outline and our introduction will see the components of the hemostasis system, the blood vessel wall in hemostasis, anticoagulant functions of intact vessel wall, procoagulant function of damaged blood vessel wall, fibrinolytic functions of the blood vessel wall. So these are some of the things we'll discuss in this lecture. We want to have an overview of the hemostatic system first. Then our learning objective at the end of this lecture, you'll be able to list the systems that interact to provide hemostasis, describe the properties of the blood vessel wall in the initiation and regulation of hemostasis and fibrinolysis. Okay, so this is a part of the introductory lecture, as I said, to hemostasis. Okay, what makes the hemostasis system significant? You may ask as why is the hemostatic system important? Why is the hemostatic system important? Why should we study it? Why should we regard it? Why should we study it? Why should we pay attention to the hemostatic system? We find out that the hemostatic system is responsible to maintain the blood in fluid state while it remains circulating within the vascular system. Okay. Making sure that the blood that moves inside our system is not clumped or clotted. It is the hemostatic system of the body that ensures that our blood, while it circulates inside our vascular system, remains fluid and not clotted. The hemostatic system is also important to maintain the integrity of the vessel wall. Maintaining the integrity of the vessel wall and make sure that the vessel wall through which the blood is moving are not compromised, okay, that they are smooth and contribute to the blood flow. Okay, the hemostasis system is also important to arrest bleeding at the site of injury or blood loss by the formation of hemostatic plug. In there is a cut or an injury to the body and the blood vessels are damaged, it is also the responsibility of the hemostatic system to ensure that this bleeding stops. Okay? Then also the hemostatic system ensures the removal of plug when healing is complete. So as clotting takes place during a process of injury or healing, it is also the hemostatic system that is responsible for remodeling, removing the plugs so that they will not also block the vessel wall. So these are some of the significance or importance of the hemostatic system. That's how they contribute to the normal physiology of the blood of the body. So by definition, we can say the hemostatic system is a complex, finely tuned interaction of cellular and plasmic blood components with the vessel wall to ensure blood flow in a fluid state. Okay? And various activating and inhibitory feedback or feed-forward system limit and regulate clot formation. Okay, so the hemostatic system comprises all the systems that are involved in the interaction of the cellular, such as the platelets that are involved in the coagulation, and the plasmic blood components, that is the factors that are also involved in blood clotting, with the vessel wall to ensure blood flow in fluid system. Okay, so that is what we have as the definition of the hemostatic system. Then this hemostatic system have components of the hemostatic system. The 5 major components of the hemostatic system include the blood vessels, the platelets, the coagulation factors, coagulation inhibitors, and the fibrinolytic elements. Okay, and the fibrinolytic elements. These are the components that participates in the hemostatic system of the body. So let's look at the blood vessel wall as part of the components of the hemostatic system. The structure of the blood vessel can be broken down into three layers: the intima, the media, and the adventitia or the externa. Okay, these are the structure of the blood vessels are partitioned into the intima, media, and adventitia. The intima is the innermost layer. Okay, when you look at the blood vessel here, and look at the intima, is the innermost layer, and the surface is covered with single layer of endothelial cells, which we can see here. This is the intima, the innermost layer, and is covered with a single layer of the endothelial cells. Supporting the endothelial cell is the internal elastic lamina composed of elastin and collagen. Okay, subendothelial connective tissue is composed of collagen and fibroblasts in the vein, and collagen and fibroblasts and smooth muscle cells in arteries. Okay, in the veins, the subendothelial connective tissue are composed of the collagen and the fibroblast, while in the arteries, the subendothelial connective tissue are composed of the collagen, the fibroblast, and the smooth muscle cells. The media or the middle layer contains mainly circularly arranged smooth muscle cells. Smooth muscle cells, that is, we find them in the middle layer or the tunica media. The media or middle layer contains mainly circularly arranged smooth muscle cells. It also contains collagenous fibrils. It is separated from the adventitia by external elastic lamina, as you can see in the diagram. The muscle cells contract and relax, whereas the elastins allows vessels to stretch and recoil. The adventitia, the outermost layer of the blood vessel, is composed of collagen fibers. You have collagen fibers and fibroblasts. This fibroblast protects the blood vessels and anchors it to the surrounding structures. So this is the structure of the blood vessel wall. Why we pick the blood vessel wall? Because what we are going to be discussing, the blood flow we are going to be discussing, happening inside the blood vessel wall. So we take time to look at the structure of the blood vessel walls and their also position. That is very necessary. You should know that. Okay. Now, from the blood vessels wall, we are still zeroing down. We go down to the endothelium. Okay, we go down to the endothelium. The endothelial cells, as you know, are also complex. They are complex, heterogeneous. They are heterogeneous. They are complex and they are heterogeneous. They are distributed throughout the body. See, they display unique structural and functional characteristics depending on their environment and physiologic requirements, not only in subsets of blood vessels such as artery vessels, veins, but also in the various tissues and organs in the body. Okay, so the endothelial cells are a complex set of cells and heterogeneous. They adapt depending on the function they are going to perform in the body and the location where they are located in the body. And these endothelial cells, as we can see, they function in so many, a multitude of physiological processes, including the control of cellular trafficking, the regulation of vasomotor tune, and the maintenance of blood fluidity, which is where we are much interested in. So the endothelial cells form a smooth, unbroken surface that causes the fluid passage of blood. The endothelium functions in a multitude of physiological processes, including control of cellular trafficking, say that, regulation of vasomotor tune, and then maintenance of blood fluidity. Okay, and that is where we are going to concentrate. The function of the endothelial cells we are going to discuss is its ability to maintain the blood in fluid state as it remains circulating in the body. That is our discourse on this. So before we start going into the normal function of the endothelial cells, we have to take time to look at the normal intact endothelial cells. Okay, the normal intact endothelial cells. Okay, so so normally the endothelial cell, we say the endothelium represents a dynamic interface between flowing blood and the vessel wall and produce a lot of factors that regulate blood fluidity. Okay, so the endothelial cell, normally the intact vascular endothelium prevents thrombosis. Okay, it prevents thrombosis, does not allow the blood to clot by inhibiting platelet aggregation, preventing coagulation activation and propagation, and enhancing fibrinolysis. Okay. So the endothelial cells, as we know it, does not support the blood to clot due to some of the factors, some of the secretions that it can produce, synthesize. Like we know, the endothelial cells synthesize and secrete a variety of substances that maintain normal flow, like the prostacyclin is a platelet inhibitor and a vasodilator. Okay, is synthesized through the eicosanoid pathway and prevent unnecessary or undesirable platelet activation in intact vessels. Okay, the endothelial cells also secrete nitric oxide. It's synthesized in the endothelial cells, vascular smooth muscle cells, neutrophils, and macrophages. It induces smooth muscle relaxation, subsequent vasodilation, inhibits platelet activation, and promotes angiogenesis and healthy arterioles. It also secretes the tissue factor pathway inhibitor, TFPI. The endothelial cell produce this anticoagulant, TFPI, which controls activation of the tissue factor pathway, also called the extrinsic coagulation pathway. We also secrete thrombomodulin. Okay, it synthesize and express this substance on their surfaces, inhibitors of thrombin formation. Okay, facilitated by endothelial protein C receptors as a cofactor and heparin sulfate. Okay, EPCR binds protein C and thrombomodulin catalyzes the activation of the protein C pathway. Protein C pathway downregulates coagulation by digesting activated factors 5 and 8, thereby inhibiting thrombin formation. Heparin sulfate is also synthesized by the endothelial cells and is a protease inhibitor. So what this slide is trying to let us know is that to maintain the blood in fluid state, the endothelial cells secretes some substances which we have gone through: prostacyclin, nitric oxide, TFPI, thrombomodulin, heparin sulfate. And all these things will work towards ensuring that blood does not clot unnecessarily, that blood that flows in the system remains fluid. So this is the basic function of the endothelial system, the endothelial cells. Also, apart from these substances that the endothelial cells also secretes. Even the shape of the endothelial cells, the way the endothelial cells are organized in the blood vessels, like their shape, they are rhomboid and continuous cells. They are rhomboid and they are continuous. This structural arrangement of the endothelial cells provides the smooth inner surface of the blood vessels that prevents harmful turbulence that otherwise may activate platelets and coagulation enzymes. So the structural arrangements of the endothelial cells also ensures that blood flows in a fluid cells, in a fluid form. Also, the endothelial cells form a physical barrier, okay, separating procoagulant protein and platelets in blood from collagen in the internal elastic lamina that promotes platelet adhesion, the tissue factor in the fibroblasts and smooth muscle cells activate coagulation. So the endothelial cells also form a barrier between the blood and the subendothelium, because when you go down to the subendothelium is thrombogenic. It promotes what? Thrombosis, the substances that are released from the subendothelium. So endothelial cells, in trying also to maintain the blood in a fluid state, also forms a physical barrier between the blood and the subendothelial substances. Before you now add, it has physical contribution to maintaining the blood in fluid states by the way it's arranged and its shape, and also forming physical barrier, and also by the substances it secretes. So that is how the endothelial cells contributes to ensure that the blood remains in fluid states as it circulates in the body. Okay. So we are going to look at the properties of the endothelial cells one on one. So we have the anticoagulant or inhibitory property of the endothelial cells. So the endothelial cell suppress clotting through the production of this. We mentioned nitric oxide, prostacyclin, heparan sulfate, thrombomodulin, tissue factor pathway inhibitor. Okay, inhibition of platelet functions and coagulation by several endothelial molecules. Endothelial cells expresses nitric oxide, prostacyclin, and ectonucleoside triphosphate diphosphohydrolase, which inhibit platelet adhesion and aggregation. Okay, secretes heparin-like molecules expressed on endothelial surfaces, serve as cofactor for antithrombin 3, inactivating several coagulation factors. Endothelial cell expresses tissue factor pathway inhibitor, which limits the action of tissue factor and inhibits excessive tissue factor mediated activation. We talked about the thrombomodulin. So in trying to um, give in trying to contribute to the one of his properties of anticoagulants or inhibitory property of the endothelial cells, these substances that are being secreted contribute to making the endothelial cells maintain the blood in fluid state. As we can see from the diagram, normal blood flow in intact vessels. Okay, red blood cells and platelets flow near the center. White blood cell migrates and roll. Endothelial cells and the ECM provide several properties that suppress hemostasis. Endothelial cells, okay. So this the same diagram depicting the function of the endothelial cells, how the endothelial cells properties contributes in making the blood remain fluid as it circulates in the body. All this nitric oxide, prostacyclin, and make sure that these platelets don't aggregate. Okay, ATI3 and heparin inhibiting coagulation, TFPI stopping the extrinsic pathway of coagulation, TM and thrombin and protein C as cofactors, inhibiting activated factor 5 and factor 8, which participate in coagulation and the by one, and the uh, prostaglandin and the plasmin activator inhibitor also plays parts in fibrinolysis. So these are some of the functions of the endothelial cells as per anticoagulants or inhibitory property of the endothelial cells. Okay. So we also have another property of the endothelial cells, which is the procoagulant property of the endothelial cells. Anticoagulant, procoagulant. Although the intact endothelium has anticoagulant properties, the vascular intima promotes coagulation when damaged. Okay. When damaged, that vascular intima that we say that maintains blood in fluid state promotes what? Coagulation when it damages. Firstly, any harmful local stimulus, whether mechanical or chemical, induces vasoconstriction in arteries and arterioles. Okay, smooth muscle cells contract, the vascular lumen narrows or closes, and blood flows to the injured site is minimized. The blood that flows to the injured site minimized. This is how the blood vessels, okay, tends to stop bleeding. Although veins and capillaries do not have smooth muscle cells, bleeding into surrounding tissues creates extravascular pressure on the blood vessel, effectively minimizing the escape of blood. So this is the initial response when the blood vessel is damaged. Any harmful substances that damage the blood vessel, this is the initial response, induces vasoconstriction in arteries and veins, and arterioles, smooth muscle cell contracts. So that is the initial response we will have. So after that initial response, endothelial cells, adhesion molecules. Okay, so the subendothelial connective tissue of arteries and veins are rich in collagen, a flexible, elastic, and structural protein that binds and activates platelets. Okay, so during the course of vascular damage, the subendothelial cells are exposed, and the collagen that contain there are exposed, and the collagen also promotes and activates platelets. So when the subendothelial cells are exposed, by implication, the collagens and then platelets activations. So at the site of vascular injuries, von Willebrand factor from storage von Willebrand factor from storage sites called Weber palette body, when activated by vasoactive agents such as thrombin. Von Willebrand factor is a large multimeric protein that is necessary for platelet to adhere to exposed subendothelial collagen in arterioles. So when the subendothelium is exposed, the von Willebrand factors from the Weber palette bodies of the endothelial cells are exposed, and they help the platelets to bind to collagen. So von Willebrand factors captures platelets from the circulation via the interaction of von Willebrand factor glycoprotein 1b on the platelets. Okay, the binding of the glycoprotein 6 with collagen leads to further platelet activation. This results in the integrin-mediated adhesion of platelets and cellular activation, releasing mediators. So when the subendothelial cells by implication is exposed, by implication the collagens there are exposed, and von Willebrand factors are also being released from the Weber palette bodies of the endothelial cells, binds with von Willebrand factor binds with the collagen and also binds with the platelets, binds with the collagen with glycoprotein 6, binds with the platelets with glycoprotein 1b receptors on the platelets. So this binding of the platelets to the von Willebrand factor leads to the activation or the exposure of the integrin-mediated adhesion platelets receptor, and it will lead to platelet aggregation, further give rise to platelet aggregation. Also, thrombin binds to glycoprotein 1b and activates protease activated receptor 1 and receptor 4. Okay, agonist stimulation results in integrin activation and the transition to an active conformation which binds fibrinogen and mediates platelets aggregation. We'll still see this description when we go further, to the okay. So that is the procoagulant property of the endothelial cells. In the event of any vascular damage, the subendothelial cells become exposed. Okay. Okay. All right. So we also have the fibrinolytic property of the endothelial cells. Okay. Okay. But before that, okay. Okay. So the endothelial cells also supports fibrinolysis. That is the removal of fibrin. The removal of fibrin to restore vessel patency. By secreting tissue plasminogen activator during thrombus, by secreting tissue plasminogen activator. That is how the endothelial cells support fibrinolysis, because it secretes tissue plasminogen activator. During thrombus formation, both the TPA and plasminogen bind to polymerize fibrin. TPA activates fibrinolysis by converting plasminogen to plasmin, which gradually digests fibrin and restores blood flow. Okay. The endothelial cells also regulate fibrinolysis by providing inhibitors to prevent excessive plasmin generation. Endothelial cells, as well as other cells, secrete plasminogen activator inhibitor, a TPA control proteins that inhibit plasmin generation and fibrinolysis. Another inhibitor of plasmin generation, thrombin activatable fibrinolysis inhibitor, is activated by thrombin bound to endothelial cell membrane thrombomodulin. So these are some of the substances that are being secreted by the endothelial cells that helps the endothelial cells to function in this fibrinolytic property, ensuring that vessel patency is being restored. The smoothness of the vessel is being restored after healing, after any healing. Okay, so do you have any questions? Do you have any questions?